Hello viewers, today we will discuss the basic idea of paleontology, evolution and origin of life. Introduction to paleontology. Paleontology by simple means is the study of the biology of rocks and is derived from a Greek word that can be splitted into paleos means past, ontos life and logos study. It can be thus derived that paleontology is the study of past life, and anything that gives the evidence of past life is called as fossil, that contains only a tiny fraction of the past life. The reason can be attributed to the fact that we do not actually know how many species existed in the geological past, because all of them were not preservable due to a lack of hard parts, and those with hard biological structures were not necessarily preserved. In addition, this stratigraphic record is bounded by unconformities, and there are more gaps than records. Hence, our understanding about the history of life is extremely feeble. We can, however, make some reasonable interpretation on the origin and extinction of species. The process of fossilization is essentially a journey of an animal or plant from biosphere to lithosphere, and two conditions are necessary for a life form to become a fossil. First, it must possess a skeleton of some kind or other, because soft parts are readily decomposed. Consequently, such animals leave no trace of their existence except its mere imprint in a material plane, or else it will be shattered into pieces. The following are the main kinds of preservation. Number 1, the entire organism might be preserved occasionally, including soft parts and skeleton. Woolly mammoth and rhinoceros found preserved in the ice and muds of northern Siberia are the best instances. Additionally, some insects also get preserved in the fossil resin or amber. The skeleton sometimes may also get preserved almost unchanged. It remains in its original condition except that it has lost the organic matter. For example, shells in the Pliocene beds of England. In some plants and in animals with chitinous skeleton such as grotolites, the organic material becomes carbonized. The volatiles including oxygen and nitrogen are lost, and thus the relative percentage of carbon increases. Sometimes the skeleton disappears entirely, leaving behind mold, especially when the skeleton consists of aragonite and is embedded in a porous stratum. When the shell is covered up by sediment and the soft parts have been decomposed, the inner part is filled with the same material. The carbonic acid in percolating water dissolves the shell. This structure so formed is called as mold, which is called as external or internal depending on the surface it represents. In some fossils, the minute structures as well as the form of the organism is revealed, but the original material of the skeleton has been replaced by another mineral. Fossil woods are the best examples of this kind, that shows cells and vessels just as in existing trees, except that the wall are made up of silica instead of cellulose. This process is called as petrifaction and is considered as the best mode of fossilization. The footprints of animals and the impression of jellyfishes are sometimes found in the rocks, and the fossils of this type are known as imprints or trace fossils. Sometimes they are also called as ichnofossils, and their study is known as ichnology. Paleobiology is defined as the study of life that existed in the geological past. The information of past life comes from the study of fossils, and paleobiology can be considered as a kind of an interpretation. Coming to the paleoecology. Ecology is the scientific study of the way in which modern organisms interact with their environment. In practice, ecologists study a very wide range of questions, but there are two main branches of this study: autoecology and synecology. Autoecology is concerned with the ecology of an individual taxon, such as cheetah or blue mussel. Questions tackled might concern mode of life, feeding and breeding habits, and physical and chemical factors that limit distribution. Synecology examines the complex interaction between population of a single species or between communities of organisms. Examples of synecological studies might concern the relationship between a variety of big cats and their prey on the plains of Africa, the organisms associated with the mussel patch on a rocky shore, or the chemosymbiotic relationship within deep sea living communities. Examining topics such as trophic structure and diversity, paleoecologists seek to gain similar understandings about the fossilized organisms and ancient communities. Paleoecological studies may have relevance outside the sphere of paleobiology. Organisms are not randomly distributed and are adapted to a particular set of environmental conditions, for example, salinity, light levels, substrate, consistency, and temperature. Once it has been established that a particular fossil taxon had a particular set of habitat requirements, the presence or absence of that taxon may be used in a broader geological context as a paleo-environmental indicator. For example, stenohaline organisms such as echinoderms and brachiopods, which have no ability to excrete water gained by the osmosis, are limited to normal marine salinity. Thus, the presence of the body fossils of these higher taxa has been widely used as an indicator of prevailing normal salinity. For other groups, a detailed knowledge of the paleoecologies of their component genera or species has been invaluable in finally subdividing paleoenvironments. For example, relative proportions of millulid, rotulid, and textulid foraminifera in sediments give important information concerning the salinity of a depositional environment from brackish to hypersaline. Brackish water are dominated by rotulids and textularids, whereas millulids and rotulids make up the dominant species in areas of normal or elevated salinity. In terrestrial sediments, details of the paleoecology of plants, such as the relationship between leaf shape and temperature and between concentration of stomata and atmospheric carbon dioxide levels, may be used as paleoenvironmental reconstruction. Most recently, there has been an added interest in environmental change, for example during glacial periods or after mass extinctions. It may give useful clues as to how it is linked to human activities. The theory of evolution by natural selection, put forward by Darwin in 1859, is the greatest unifying theory of the natural selection and theory of biology and paleontology. In this context, evolution is the change that occurs between successive populations of organisms due to their modification in response to selection pressures. The potential to change is provided by genetic variability within populations and by genetic changes through time, that is mutation. The pressure for change to occur exists outside an organism is provided by interactions within the environment. These interactions may be predominantly physical or biological effects. Small-scale changes in population giving rise to new species are defined as microevolution. Larger-scale changes, such as the origin of new higher taxa, which may have new body plans or new organs, are defined as macroevolution. The study of evolution also includes the study of patterns of diversification and extinction. Microevolution may be the end result of microevolution working over a long time scale, or it may be a suite of emergent properties that require unique interpretations. Theoretically, it should be possible to study aspects of the morphologies of fossils collected bed by bed throughout a rock sequence in order to elucidate patterns of evolution. At some scale, all deposition is intermittent, and this means that there are gaps of some scale in all narratives retrieved from the fossil record. And research has generally been concentrated on these locations. Coming to the microevolution, the set of potentially interbreeding members of a population forms a species, which contains a range of variation in its appearance, the phenotype, and the definition of a species must be based on the clusters of phenotypic characters which are taken as proxies for the potential to interbreed. Natural selection acts on a set of individuals so that the physical characteristics of the group and the underlying genotypes change over time. The process eventually gives rise to new species and is known as microevolution. Biologists class only gene shift within population as microevolution and define anything larger, including the appearance of new species, as microevolution. Sometimes a species gradually changes through time until the point comes where the fossil representatives of successive populations are recognized as a different species. However, a parent species often splits into more than one offspring species or evolves into an offspring species that coexists with the parent species for some time. In this case, the original population must split into two or more subsets that cannot interbreed with one another. The two best known methods of achieving this are called as allopatric speciation and sympatric speciation. In allopatric speciation events, a single original population is split into two geographically isolated elements, although it seems intuitively obvious that populations that become physically dissimilar will eventually be unable to produce offspring. The genetic basis for this change can be demonstrated in the laboratory but not yet fully explained. The fossils records can be used as a tool to help in the understanding of evolution and the formation of new species. It may be that evolution progresses gradually for most of the time, an idea which is known as phyletic gradualism. Gradual change is generally difficult to observe in the imperfect fossil record. It could be argued that in a less continuous sedimentary record, or one which is sampled less finely, this sequence of events would appear as a series of abrupt changes. Commonly, what is preserved is a long period where little or no change is observed, followed by the abrupt appearance of a new form. The theory of punctuated equilibrium attempts to explain this phenomenon not as the product of an imperfect fossil record, but as a common pattern of evolutionary change. This is done by applying the concept of allopatric speciation to the problem. L. Ridge Gould, who developed the idea of punctuated equilibrium, argue that most species probably arise in a small geographically isolated areas, and they may rise rapidly as they encounter new selection pressures. Although some studies seem to show a punctuated equilibrium style of evolution, others appear to show that evolution has progressed via phyletic gradualism, and a consensus has yet to emerge regarding these theories. In practice, most evolution is probably the result of a mixture of the change, partly depending on evolution has progressed via phyletic gradualism, and a consensus has yet to emerge regarding these theories. In practice, most evolution is probably the result of a mixture of punctuated and gradual periods of change, partly depending on the scale of sea level changes. Each geographically separated fragment of initial population contains only a fraction of the original genetic variation. So, it may tend towards differences from the original population without any active selection. Although this is now regarded as minor component in the formation of new species. More importantly, the different geographical regions will tend to produce different environmental stresses from those that were experienced before separation, leading to the selection of different successful characters in the separated populations. This eventually led to significant changes of form in the isolated populations, which may finally produce new species. During sympatric speciation, the emerging species share a geographical range but may become separated over time by differences in the behavior or in resource exploitation. Adaptive pressures act differently on these populations, and different characteristics will be favored, leading to a progressive change of form and eventually to reproduced isolation. At this point, a new species will have appeared. There is some doubt about the mechanism by which species first begin to diverge without becoming geographically isolated. Macroevolution is the study of all evolutionary events or effects larger than the appearance of a new species. This includes studies of long-term change in the geological record and of the emergence of new higher taxa. For example, new phyla linked to both of these topics is the difficult issue of how significant new structures or organs can evolve. Paleontology is central to this study, as it provides a measure of time and can identify the most likely dates of appearance of new characters or taxa. The single biggest and the most important argument about macroevolution is whether it is a scaled-up version of the microevolution or something different. If it is different, then those differences may be a reflection of the emergent properties of this complicated system and hence is still reliant on microevolutionary processes occurring. More controversially, it has been argued that macroevolution includes rapid and large-scale changes of form that necessitate steps that might initially produce organisms that are less successful than their ancestors. This is completely counter to Darwinian ideas of evolution. An example of these issues can be presented via a consideration of the evolution of major groups of tetrapods. All living vertebrates with pentadactyl limbs, that is mammals, reptiles, amphibians and birds, evolved from an ancestral fish, with the process beginning in the freshwater lakes and rivers in the Devonian. Since then, a wide variety of adaptations have appeared in these higher groups such as feather, fur and wings. It can be convincingly demonstrated that some lineages or evolving lines acquired these characteristics gradually by a microevolutionary process. Looking at the history of life, it is clear that there have been periods of major increases in diversity and periods of major innovation. Significant increases in diversity tend to happen after mass extinction and are called as evolutionary radiations. Empty niches created by the extinction events are quickly filled as organisms radiate to form new species that are able to exploit the available resources. Evolutionary radiations may also be facilitated by the appearance of major innovations, such as the evolution of hard parts by a variety of different taxa close to the Precambrian-Cambrian boundary. The two are not necessarily coupled. For example, eukaryotes, the complicated internally divided cells with which we are most familiar, evolved more than 2 gigaannum ago, possibly much more, but they did not become widespread or common until much later and around 1 gigaannum ago. They could not radiate until there was an adequate population. Species-level selection favors species that have lower extinction rate and higher origination rates than their competitors. In the long term, these species become more abundant at the expense of their less successful competitors. The pattern of species extinction would be expected to be one of the increasing chance of extinction with species age. But in some cases, this does not seem to be so. Instead, species age does not appear to correlate with the likelihood of extinction. When Valley has used this observation, which is itself somewhat contentious, to suggest a novel hypothesis for microevolutionary patterns. He suggested that competition for resources produces a dynamic equilibrium between species, in which each will continue to evolve in order to survive. This is the core of the Red Queen hypothesis, which suggests that organisms evolve to keep their biological place, or, to paraphrase the quotation from Alice in Alice through the Looking Glass, they run to keep still. The characters that help organisms to survive at times of low extinction rate may be different from those that mark survival of the mass extinction more likely. In other words, the criteria by which species are selected may vary with extinction rate. Specialist species tend to have greater survival potential at times when the extinction rates are low, and reduced survival potential when the extinction rates are high. In addition, it has been suggested that the small species will have greater chance of surviving mass extinction than larger species. So the overall trend in evolution is clearly not towards a smaller species. Establishing the nature of the processes by which life originates is one of the most fundamental unsolved scientific problems. 50 years of study has enabled us to assemble many pieces of the puzzle, but have also left a number of critical gaps in our understanding. Even the issues of when and where life began or originated are still poorly constrained. But life probably appeared on Earth between 4.2 gigaannum and 3.5 gigaannum ago. There is good evidence that the current DNA, RNA, protein basis for life developed from a stage known as RNA world, in which RNA performed the information storage and catalytic roles currently performed by DNA and proteins. Direct information of the RNA world from prebiotic chemistry seems unlikely. So much current research focuses on the possible precursors to the RNA world. Various suggestions for pre-RNA worlds have been made, but none has proved entirely satisfactory. A number of terrestrial and extraterrestrial processes can produce prebiotic organic molecules, but there are still problems in obtaining the right molecules in sufficient quantities. Development of ideas on the origin of life, the idea that all the life on Earth had a common origin, became well established only in the 20th century. Early ideas on the evolution of life were most clearly expressed by Lamarck, who described the process as one of the progression from simple to more complex and advanced forms. The observation that both simple and complex organisms are currently present therefore required that simple organisms are appearing even today from inorganic matter by a process of spontaneous generation, a concept that has its origins in antiquity. Thus, the idea of transformism, that is evolution, and spontaneous generation were closely linked in the early 19th century and put Lamarck and his followers in conflict with the religious and scientific establishment, who argued for the immutability of species, which were divinely created on the current forms. Lamarck's ideas were criticized by scientists such as Cuvier and later by Pasteur, who carried out his famous swan neck flask experiment to discredit the idea of spontaneous generation. Darwin's theory of evolution by natural selection opened the way for the modern view of the origin of life. In the Darwin's theory, there is not necessarily a ladder of progress from simple to more complex forms. Simple forms can be evolutionarily successful as complex ones. This allowed the idea that all life, simple and complex, had a single origin in the distant past, while the subject of the origin of life is hardly mentioned in Darwin's publishing writings. The following quote from a letter to his botanist friend Joseph Hooker gives us an inkling of his thoughts on the subjects at that time. And oh, what a big if we could conceive in some warm little pond with all sorts of ammonia and phosphoric acid or phosphoric salts, light, heat, electricity present, then a protein compound was chemically formed, ready to undergo still more complex changes. Oparin and Haldane in the early 20th century developed the idea that chemical reactions on the early Earth could have led to the production of a range of organic compounds forming a primordial soup in which the required building blocks of life would have been present. But it was not until 1953 that these ideas received experimental support. Stanley Miller, then a graduate student working in the famous laboratory of Harold Urey, set up his famous experiment in which electrical discharges were passed through a mixture of gases including methane, ammonia, hydrogen and water vapor, simulating a thunderstorm on the primitive Earth. The experiment produced a mixture of several amino acids, the building blocks of proteins. Miller speculated that this was how organic compound had been made on the early Earth. In the same year, Crick and Watson published their structure for DNA, the first step in elucidating the fundamental molecular basis of life. These two discoveries mean that we had both a plausible way of generating simple organic building blocks and an understanding of the macromolecules on which life depends. A detailed experimental and theoretical study of the origin of life became possible.